Greetings, everybody. Good morning, good afternoon, good evening from wherever you are connecting. Welcome to Biomeria's webinar dedicated to molecular innovations for food pathogen and maybe even a bit beyond pathogen testing and some of the latest developments from Biomeria as such. So it's my pleasure to welcome you on board today, and I shall be your host. Who am I? I am Robert Butler. I'm the marketing manager for Biomeria's food safety and quality business at Northern European level. And it's my pleasure to be with you today and to indeed be your host and moderator. But I'm joined by, perhaps more importantly, my good friend and colleague, Jean-Philippe Tournière. Jean-Philippe, perhaps I can invite you also to make an introduction to our intrepid crowd.
Yeah, thank you, hello everyone. It's really a pleasure to be able to present you all innovation at Biomeria. I'm the global manager for GeneUp Solution, and it's really today the center and really a key tool to drive innovation in factory, manufacturing site, and in laboratory.
Wonderful. So hopefully we'll take about 45 minutes of your time today. So thank you very much indeed for your attention. What I would point out is you will have the opportunity to pose your questions. If you can have a look at the hopefully top right-hand corner of your screen, you'll be able to see the text box with the question mark in there. If you click on that, it should bring up a sidebar for all of your question needs. So please do not hesitate if you have any questions during the course of the presentations and the discussion to put your questions in, and indeed at the end, after hopefully about 30 minutes or so, we will recover all of those questions and do our very best to address them for you. And if any question is not addressed during the course of that, then we will absolutely endeavour to follow up with you and come back with any extra information that you've requested. So with that in mind, let us begin. So molecular innovations from Biomeria. So we'll have a big focus on our PCR platform, but before we get to that, I'd like to have a quick overview of what we call augmented diagnostics that's from Biomeria, perhaps the most interesting of the innovations that we've been working on over the last years and will continue to develop. Augmented diagnostics is really taking the diagnostics concept for food testing, for pathogens, spoilers, and everything else in between, to that next level. It's really designed as a suite of solutions to give food manufacturers and food developers more information about the microbiological environment that they encounter all across the manufacturing and added value process involved. So it's designed to give you a smarter and more dynamic risk assessment. As such, it's designed to give you more information sooner so that you can move from what are test results after the fact, typically, more to what we call actionable insights, so giving you more information so you can take decisions sooner, act sooner, and even perhaps start to anticipate problems before they occur. And of course, ultimately, be providing safe and good quality products for the end consumer. And it's an important point because Biomeria is very familiar at the laboratory level, and perhaps we're quite well known and recognized even at the laboratory level. But the augmented diagnostics concept really transcends just the laboratory. It's really looking at that entire manufacturing chain as such, all the way from raw materials in, the manufacturing, literally the production floor itself, as well as the post-production testing and pre-release testing that goes on. So ultimately, it's designed to identify the types of pain points that are typical at the manufacturing level and can cause considerable problems at the manufacturing level as such. So really understanding the microbial flora that you will typically be encountering. Are you seeing certain pathogens or spoilers that crop up? Are they the same type of thing? If you have a Listeria mono, for example, you see that at a manufacturing level. Perhaps you see it elsewhere. Is it the same strain? Is it related? Does it have some kind of resistance to the antimicrobial chemicals that are being used and so on? These are the types of insights that this this technology and this know-how is able to bring in order to improve the general environmental monitoring process, improve that understanding, improve the rollout, and in that way improve operational efficiency as such. And likewise, as with everything nowadays, data is everything. So it's designed to be plugged into that industry 4.0 concept where all things are speaking to one another and transmitting data in real time, and that real digital transformation that we're working towards. And last quick word from me, just that overview of what that augmented portfolio ultimately looks like. So as I've said previously, bioMérieux is quite well known for our test results approach. So for whether it's sample media preparation, our BioBall standardized strains for QC testing and cross-contamination detection, whether it's enumeration and quality indicator testing in the form of our Chemunex D-count flow cytometry or Tempo enumeration, automated enumeration platform, or indeed pathogen and spoilage detection in the form of our VIDAS Cube immunoassay platform, which has been the bedrock of our pathogen business for some time, but now more and more so with the advent of GeneUp, which is our real-time PCR platform. However, augmented diagnostics is really moving us towards these actionable insights. So on the right-hand side of the screen there, where we see pathogen and spoilage mapping and data services, but perhaps more so concepts like GeneUp Typer, which we'll touch on today, the XPro program, which again we'll touch on, and indeed the connectivity aspect. And with that in mind, I think we can move on to a bit of an icebreaker for you guys in the audience. So just to warm you guys up, if we can have the first question on the screen, please. Hopefully you can all see this question. So a question for you guys, please, fingers on buzzers. How familiar are you with molecular methods for detection? Be that PCR, be it LAMP, be it anything in between, isothermal technologies and so on. Please let us know what your level of knowledge is, and be honest, because we're trying to gauge and pitch to all levels as such. So it's very interesting for us to know exactly where you feel you are at. And I'll give just a few more seconds. I can see those results coming in live. And just 5 more seconds. Very good, looks fairly static now. Okay, let's perhaps end the poll there. And unfortunately, I think I killed that, but we saw a certain level of limited practical experience, let's say, unsurprisingly being about half of people voting around limited practical experience. So good level of knowledge, but perhaps more to learn. And that leaves us in good stead for where we are today. Jean-Philippe, I think, if it's all right with you, I'd like to hand over to you.
Thank you. I can't share my screen, huh? Okay. Go. Okay. So thank you, Rob, for this introduction. So from what I've seen, most of you already heard about a molecular solution and are familiar with PCR technology. So the GeneUp, I will present you today the GeneUp augmented ecosystem, which is a combination of technology expertise in different domains, in instrument, software, PCR development, PCR system development, really, and all linked elements around microbiology, data analysis, and confirmation step. So I will focus more on the pathogen side, which is really the key and the earth of our solution, and really addressing all the main market segment and matrices. I have a very brief overview of the spoiler part, which is mainly addressed to beverage applications for alcoholic and non-alcoholic beverage. all virus testing. I apologize for my accent, but we had some pretty serious crises linked to oyster consumption during Christmas time, and we have tools to really perform some testing in this field. And we'll go to very innovative and application, combining molecular solution, data analysis, and regarding strain typing, and also the development of custom assay, which is today really successful for very specific problematic at the industry level. So at the end, we have one equipment really at the heart of a lot of application, more than 30 applications today. Okay, what are the GNUP innovations? So first of all, really, it's a real-time thermal cycler that has a very scalable throughput that is very flexible, can go from 1 to 96 tests at a time. It's a very powerful instrument, and that helps really to reduce the processing time to results, and time to answer, to get actionable action, to get actions on the industry, on the manufacturing line. So really, the goal today, it's really to reduce the time to results, to save cost, and get quicker action on the field. As well, we make a lot of efforts in harmonizing the workflow, the enrichment step, the PCR step, and as well the confirmation level, and combining different expertise at different level to really provide added value to final users. One very brief overview at the chemistry level. So we are combining not only the specificity of the PCR, but using Fred probes, primers, targeting or at the species level, the serotype level, the genus level, and combining with this approach with the MELPIC can bring a really added value in terms of interpretation and refine the analysis, and consolidate the robustness of the answer, and reach the optimum in terms of accuracy of the results. That's really to drive sensitivity and accuracy. The GNUP workflow. So this is where we bring most of, this can really highlight all the innovation that will bring in 2024 bioMérieux level. So first of all, we invested a lot in the eco-packaging, so environmental protection by bringing new packaging on the field, and we have continuous efforts in this direction to really study since the beginning the environmental impact that our solution will have during all their life. At the sample preparation level, we have a complete offer, and we have also a full expertise at the enrichment level, bringing high technology in what we could think to be classical media, but improving the recovery of stress cells, really reducing the enrichment time, for example, for Listeria application, really bringing added value to a final user, and securing the results and bringing safety and quality of the results. In terms of innovation as well, you may have heard about the 5 minutes extraction step we have with our lysis tubes and magic caps that avoid the user to open or close tubes, reducing the risk to get cross-contamination or even avoiding them totally. And the mechanical lysis is very powerful and demonstrating very good performance in all the certification bodies and international validation level. At the amplification and detection level, we are, of course, bringing the fret and melting curve technology as well today, well, today and in the coming months, we'll launch a new primary packaging of our solution with a color-coded PCR plate that will allow you to have a better and a more user-friendly experience. Also reducing the risk of mistake, improving the ease of use at the operator's level, and getting even better securized results. Everything combined with a very intuitive software that provides final results, final interpretation with directly with no gray zone, I would say. So definitely improvement in the ease of use in the coming weeks and months. And this corresponds really to a global and continuous improvement in our solution. As well, we invest a lot in very innovative chromogenic media. I can't tell you right now more, but you'll get probably more information by the end of the year for new application and with really fantastic added value. And by the way, I would like to thank the R&D team that I dream about some of those solutions since more than 10 years, and you made it. So again, one of the key elements, it's really to provide added value to the industry, trying to improve the productivity, securing the results of the labs, and trying to bring added value to the full operational chain. And one of the elements, it's really to reduce the time to results, the enrichment time for food pathogen detection. The eGenap Easy Prep really brings innovation combined with nucleosens technology. It helps us to really concentrate the target, removing potential PCR inhibitors, and it helps to increase the sensitivity of the method by something like 200 factor, allowing us to bring really added value for some specific application. In this case, for cocoa industry, that will really help them to have the same shift work. So working on a new batch of products and getting the analytical results by the same day and at the end of the shift. So this provides tremendous added value to the global chain, not only at the laboratory level, but also at the full logistic level, full chain, saving tremendous cost at the end of the day. Again, we'll develop this technology. We're about to launch it for some specific application. It will be used as well for virus detection and getting from a manual sample preparation to a semi-automated mode. Here, for example, for cocoa industry, we'll get fully validated results in less than 12 hours. So again, I'm bringing extra value to operators. Globally, we have a full range of parameters, providing complete solution for all the main segments available today of concern. Depending of the territory, we may have some slight adaptation of the parameters. So we are really well equipped to fulfill expectation of the dairy segment with different approach, combining Chronobacter, Salmonella, and very soon we'll launch the Enterobacteriaceae parameter. In produce segment, also addressing different testing expectation for Salmonella, for EEC, and pathogenic E. coli, addressing really the virulence gene to refine the diagnostic of the methods and reduce the positive presumptive rate. As well, we have a unique solution for virus detection. I'll come back slightly after, mainly in red fruit, red berries. And we have a full range of solutions as well for the animal protein segment, between the different Salmonella, serotype Salmonella species, detection in primary production, Salmonella enteritidis and different serotypes ongoing on development currently, as well with a Salmonella quantification approach, mainly in poultry industry, pork industry, to really help the industry to find, to assess the right level of safety. Seafood segment as well, a full list of application linked to virus, and processed food segment as well with chronobacter and different solution as well. Okay. So we have also even more recent and more innovative solution trying to fulfill some additional expectation from the field. First of all, the Nutraplex Pro, combining E. coli, staph, and Salmonella detection in one single PCR using one universal enrichment broth. Cannabis range that is more applicable on the North America territory. And also we plan to, we have currently AOAC validation for multiplex detection of Salmonella and Listeria on environmental surfaces. So currently validated on North America through the AOAC, but we are currently investigating some potential other application on other territories. Again, the food virus application, with also some detection of norovirus, hepatitis, using mangovirus as a control. And we are really currently offering a good solution to provide tools for seafood safety and red berries application and berries application as well. So most of our solutions are validated by international certification body at different level or at the AOAC level and at the ISO level for all our food pathogen testing and for environmental testing, primary production. So we have really a complete, and I think we have one of the largest scope of validated matrices with more than 250 matrices validated at AOAC and multiple ISO categories as well. So it's a significant investment in method validation and method verification upstream in R&D. So globally, we target as well multiple sample size from 1 to 375 grams, really to reach, to streamline and to optimize the analytical process and really to provide added value and save time and cost to the final users. Also, we are working on different innovative approach, working on different dilution ratio, and as well for meat processors using microtally at North American level. Okay, I think we'll now switch to another poll. So speaking about even more innovative approach, and so I think I can give the stage to Rob. Are you around?
Yep. Certainly, yeah, I see people, quick fingers out there already answering the question. But yeah, poll number 2. So is your organization currently doing any form of strain typing or characterization, regardless of the technology, whether that's PFGE, whole genome sequencing, next gen genome sequencing, ribotyping, MLST, 16S, or anything in between? Are you involved, or have you been involved in any of that type of activity as such? Is any form of routine at a manufacturing level? So we have, yes, we're doing this in-house routinely, or yes, we outsource, yes, but we outsource this to some other expert laboratory or contract lab service. No, but I am curious about the benefits and I'm interested perhaps to look at this, or no, I don't really understand this type of technology and I'm not sure how it fits into my organization. So interesting. Just maybe a couple more seconds just to get those last answers in as such. Okay, so it looks like an interesting split as such. Okay, so we have quite a bit of diversity there. So outsourcing and perhaps not so much familiarity, perhaps being the significant trends pulling forward there as such. So that is interesting. So I think that segues us quite nicely, Jean-Philippe, perhaps to the next section of the discussion around some of the capabilities of GNUP.
Yeah, very good to know, very good to know, and a good switch, good transition to the next topic today. Speaking about Typer. Sorry. Again. So this is one of the major launch of and major development of bioMérieux R&D in those past years, and really with a tremendous finality. And to me, it's a very exciting solution that will provide. First, we provide it on the service format, but we'll soon be able to provide, solution that would be able to be used at facilities level. So using the GNUP instrument as a fantastic and powerful tool combining with data analysis. So we'll provide a method equivalent in terms of performance and accuracy and to the PFG application, which is pretty old and that has been the reference during decades. And I think that to my opinion, the Typer could be a really fantastic alternative solution and very easy to use, and based on the simple PCR colony. So again, currently it's offered on the Listeria monocytogenes for now, and we have development ongoing on other parameters. But the idea is really to have a probabilistic microbial typing method, and that will help the industrial to take decision based on the if you have any positive presentive in your samples. If you have, well, first of all, if you have no positive, no problem, you release your product. If you get a positive, then you will ask yourself the question: is it the same one? Is it unused? Is it coming from my own factory, from the ingredients, the raw materials? So, and using this technology will help you to get very quickly some answers, and to get results in less than 24 hours. Really to determine the source of contamination. Is it a true contamination, or does it comes from a cross-contamination of my control strains? So the GNUP Typer will provide you a tremendous added value and flexibility in your daily activity. Really, it will be a fantastic tool as well to be in the crisis mode management, to really push deeply investigation very quickly in your manufacturing site. So the idea is really to define very quickly an efficient action plan, and where you can address all questions in less than 24 hours. So you will quickly know if it's a new strains, if it's a previously detected strains, or if you get inconclusive results, and that will be a helpful tool to fulfill current issues on your decision process. Again, this tool is currently provided as a service. There will be a complete new full launch by the end of the year, and you will keep you posted about this tremendous innovation at bioMérieux level. Okay, another big source of innovation, it's the GNUP custom assays based on the XPro program. And here it's really based on specific issues that are encountered by industrials, by laboratories that face very unexplored matter and topics. And the idea is providing really a customized system that will help you to solve your issue. And we can go from a simplex system to multiplex assays with a streamlined sample preparation. So it's a bit outside of the classical GNUP workflow. We may have to optimize some sample preparation, some enrichment for specific application, for the detection of specific molds or pathogens or spoilers. And the idea is really to provide as well a kind of predictive assays depending on the situation and targeting really novel genes for really true information with high added value. So we developed the assays. We really build partnership with industrials, and also it's a way to get very quickly on the market and to get very quickly some solution, and with a true partnership between bioMérieux and other operators. I will provide you a few number of examples that have been successful, sorry, and yeah, driving so all the industrial projects, partnering with different key player in their field of application, with body armor, targeting pro beverage bacteria, different bacteria that can spoil their product. Developing also really investing on spoiler level to really help breweries to ensure the quality of their production, to speed up the release of their batch, and really to anticipate any potential issue during the manufacturing process. As well, very promising and very targeting Alicyclobacillus and heat-resistant molds as well, that are supporting the pasteurization process, and also very impactful results for the optimization of the logistic flow at this level. Again, a new market, a new application for all nutraceutical products, developing triplex tests to get only one detection and one enrichment to get the results on 3 different parameters. And we are here at the border with pharmaceutical application, but also providing innovative solution to really help our customers to release their product in a faster mode, and to save cost and time. Okay, so at the end, the GNUP instrument is really at the center of multiple expertise at bioMérieux level. At the blue line, all the tools that are used in the lab to perform the sample preparation, the enrichment media. We have strong expertise in microbiology, chromogenic media, global and support worldwide for training, service, and a fantastic team of data analytics and innovative genetic markers. Again, tremendous work performed by our R&D team. And also a very good complementarity with VIDAS solution for some specific application where it brings really added value and good serenity in giving good results. Okay, now I think Rob will talk to you a bit more about the Connect Up and the way we manage data in laboratory.
Yeah, certainly. Thank you, Jean-Philippe. So if I perhaps just bear with me for one moment while I take back the presentation as such. So it's hopefully that, yeah, I think I am sharing. So Just a quick word on connectivity aspects. So GNAP is fully connectable as a device. We mentioned connectivity earlier. So GNAP is capable, very powerful tool in terms of the technology that it employs, but that's only as good as your data management is able to take care of how you deal with your data flow, what is going into the GNAP, what's coming out of that as such. And GNAP is indeed designed to be connected to our Connect Up platform, which you may or may not have heard of. That is essentially a data transfer tool that assists in how we set up the tests and indeed what is coming out of that GNAP to be able to push that to other LIMS systems or ERP systems that you may have. And it's really designed to limit any of those manual steps or transcription steps, or where perhaps you're using Excel to manage data that's coming out of these things. Connect Up is really designed to interface with the GNAP, but not only with the GNAP, but with all other bioMérieux suite of solutions for food safety testing, in order to traffic that information, that bidirectional flow of information, and ensure the most efficient transfer of data, the most accurate, and the greatest data fidelity as such. And if you'll bear with us, we do have a very quick video. It's just under 2 minutes long, just detailing how Connect Up can actually benefit the life with GNAP as such. Very good. Thanks indeed for your patience there. And I shall just revert back to the final stage of the presentation. So hopefully I will share the right document. So thank you for that. So just before we move on to the final step, which is the Q&A, and I see we have many questions coming in fact. So any last questions, please do put them in and we will make sure we get to them. But just on your screen there, you see for those people in Northern Europe, if you are keen to know more and you want to come and visit us, we would encourage you very much so to do so this year. So particularly in the UK and in the Benelux region, we'll be running a Future of Food Pathogen seminar, a full day, looking at the GNAP, looking at other aspects of our augmented diagnostic suite of solutions. So if you do like what you hear, a good way to get more information and more in-depth knowledge and maybe even a live demo is to join us on those days. And certainly the UK one is now set in stone, Tuesday, March 26th in the Birmingham area, location to be and venue to be communicated very, very soon. It's fully free of charge, and we would like to welcome you there indeed, as well as a couple of Camden BRI events, a webinar around ISO methodologies pertaining to the food environment, as well as alternative methods and rapid methods, and how to perhaps approach those if you are looking to adopt moving away from traditional methods. And indeed, we have a seminar coming up in July 10th, Wednesday, July 10th at Camden BRI, particularly around ultra-processed foods. So we would encourage you to join us there. So now, moving on to the Q&A. So, Jean-Philippe, I think you and I need to get a little bit busy now. So in the last 5 minutes, we'll hopefully cover as many of these questions as we can. If we do not cover your questions, I see some of them are quite specific around validations and report numbers and so on. We will absolutely endeavor to follow up with you and come back to you on a one-to-one level. So sorry if we don't cover your question today. But I see some questions, so let me just, can I pick out some of these ones? So Jean-Philippe, perhaps one for you. Does GeneUp detect dead cells as such? What about dead cell DNA as such? Is that a concern?
So that's really a long story using PCR methods, the fear to detect dead cells. So now we have very good solution available using the PMAX, so which is a intercalling agent that will avoid the detection of dead cells signal. Yeah, by chemistry action, it will inactivate all the DNA from dead cells and avoid its amplification by PCR. So removing dead cell signals from the detection by PCR. So usually we have some very specific application, or for some very specific matrices like heat process food, like I don't know, infant formula, for example, that could be subject to this kind of issue. But yes, we have a solution to avoid this the detection of dead cells.
Thank you, Jean-Philippe.
And
We have
validated, by the
few.
way. It's officially validated. Sorry.
Absolutely, yes. We had a couple of questions around that. So around things like viability dyes such as Propidium monoazide, PMA, Ethidium monoazide, EMA as such. So yes, there is that additional component with the PMAX component that can be used to manage that if that is indeed a concern. Mm-hmm. So next question is, I have here, so interesting one. Does the method have the ability to identify the whole microbial population in a sample? For example, in case of identifying the microbial contamination on a filtration membrane. So Jean-Philippe, perhaps, I don't know if you want that question or I'm happy to take it. From my perspective, probably PCR in such a case isn't probably the right tool for that when you're looking at more population identification. I think perhaps that's more a metagenomics question. So we can answer that. I think if I refer back to our augmented diagnostic suite of solution, that is absolutely something we can look at when we're looking at, let's say, a population identification spectra. But I wouldn't say that's something perhaps for PCR or indeed for GeneUp as such in such a case. Jean-Philippe, have I got that right?
Yeah, you're right. I see a lot of questions coming.
Yes, indeed.
Kind of avalanche.
There is indeed a few on typing. I think we might have to come back to those as such. We do have a question on what type of spoiler organisms does the GeneUp detect? So we did touch on that. And just to give a bit of an insight, obviously it's a quick answer, but our spoilage is particularly around the beverage type of products and also particularly in the brewing industry. So we're able to detect things like alicyclobacillus, so ACB. If you have ACB concerns, we have a solution on GeneUp that not only detects ACB, alicyclobacillus, but it also has within the assay the ability to distinguish whether it is producing the spoilage, the particular spoilage compound that is the guaiacol, that is the particular spoilage component that it's that causes problems as such. So we have both the ability to detect the ACB as well as to determine if it is a guaiacol producer or not. And I believe that is quite unique on the marketplace to do that in the same assay. There are other also bacteria and yeast detection for various multi-spoiler assays, such as lactobacillus, pediococcus, acetobacter, gluconobacter, bacillus as well. So fairly broad spectrum, particularly interesting for the beverage industry, as well as certain yeast strains, Decara, Saccharomyces, Candida, Pichia, amongst others as such. So I'm hoping that answers your question. And indeed, I know that's quite quick. If you do have other questions on that, then please do reach out to us as such. And I think we're probably coming to time, so I think we might need to perhaps one more. Can I do PCR in my lab without a separate room? That's a fairly straightforward one, Jean-Philippe.
Oh yeah, so that's more a lab organization question. So definitely you can get only one room. You just need to allocate a specific space for what we call a DNA negative area and DNA positive area. So you just need to separate properly your tools, your tips, and to avoid, of course, cross-contamination. But I've visited a lot of labs that were running everything in the same room without any issue. Of course, if you have separated rooms to perform all the different steps, it's better. You decrease the risk and you improve the quality of handling, but it's definitely not mandatory as long as soon as you follow the good laboratory practice and you pay specific attention to really avoid the cross-contamination, for sure.
Excellent.
I see one. What is the output of the typing? It's really the idea is really to provide a clustering. All your strains will be classified and regrouped and cluster to proceed like a kind of phylogenetic tree. You will definitely get more information in the coming months, I would say. But if you really need more detailed answers, please do not hesitate to contact your bioMérieux representative. Again, it's a fantastic tool coming on the market, and that will really help the daily life of most of the labs.
Excellent. Now, guys, I think we probably would take the rest of your day if we wanted to go through all of the questions. So I propose to call an end to proceedings there. For anybody that put questions, there are quite a lot of questions, perhaps more questions that we were unable to answer due to time constraints. We will absolutely follow up with you as soon as we can. In closing, I would just like to say, if you have enjoyed today's webinar, or if you haven't, or you have some suggestions to how we can improve things, you will receive a survey link immediately after the webinar. We would be very, very grateful if you could take a couple of minutes of your time just to give us some feedback over what you liked and what you would perhaps prefer to see in the future. We would be most honored if you could do us that service. And indeed, otherwise, I would just like to thank everybody for their attendance today, their excellent questions, and we hope that it's been useful to you. And it was designed really to give you just an insight into what we've been working on. It's not fully encompassing and fully detailed. So if you do wish to know more, please do come back to us and we would be happy to tell you more, or indeed show you more as much as we possibly can. So with that in mind, thank you very much indeed. Thank you, Jean-Philippe, for your excellent presenting. And thank you to the back office guys that were running the show, making the webinar move nice and smoothly in the background. Thank you, everybody. Have a good rest of the day. Have a wonderful weekend.
Thank you. Bye, everyone.